My name is Bill Clemons, so I'm the Hanisch Memorial Professor of Biochemistry at Caltech. I also have a second role. I'm at the Chan Zuckerberg Initiative, where I'm the Program Officer for Diversity.
That's really cool. And then how did you get interested in research in the first place?
So research has always been something I think I've been passionate about since I was very little. I've always been a curious person and interested in discovery and understanding how the world worked. Never been satisfied by people saying that they didn't know how something worked. And so that's been a driving force for how I've thought about problems in my life and curiosities that I've encountered. I think being curious has been a real motivating part of my life. And so I'd say as far back as I can remember, I think I've been a scientist because I don't remember a time where I wasn't asking questions and trying to take things apart to understand them.
Yeah, okay. And then in terms understandable to like a high schooler, could you give me a quick overview of your research?
So my research kind of spans a number of different areas. In summary, we're broadly described as structural biologists. And what that means is that we're interested in understanding macromolecules at atomic resolution. As most people know, the genome is organized in a way that you make genes, and those genes define, for the most part, proteins. And proteins are just polymers of amino acids, 20 amino acids that are strung together in sequences that when you make that polymer, it folds up into a three-dimensional structure that has function. While today we actually can go a long ways towards predicting what that structure looks like, understanding the details and how the mechanism and how it works is really the motivation for structural biologists. We want to understand how those structures change relative to time and space and how they interact with other molecules to do their function. And so we use tools like electron microscopy or X-ray crystallography, which are just different ways of using wavelengths of light to interact with biomolecules to then computationally develop these high resolution three-dimensional molecules where we actually can build the atoms of the polymer into a three-dimensional structure and then use that to understand how it works. It's almost like thinking about getting a blueprint for a car or house, right? If you had a blueprint that showed you all the different parts, then you can make really clear inferences about how it works.
Yeah, that's really cool. And then like what were some of your most significant findings or publications?
But a lot. So, I started my PhD working on the structure of the ribosome. And that was the sort of molecule that actually makes the polymers that form proteins, and that's universal in life. That work actually led my advisor to get the Nobel Prize. And so that was sort of, one of those highlights in a career, which is worth noting. After my PhD, I went on to do a postdoc at Harvard Med. There, we worked on my first membrane protein structure, which we call the translocon, which is involved in the secretion of proteins across biological membranes and also for the synthesis of membrane proteins. And so these are proteins that have to live in the lipid bilayer environment that surrounds a cell or forms a sort of the compartments within a eukaryotic cell. Since then, my real interest has been to continue to understand processes involved in making these special cases of proteins that exist in these membrane bilayers. They're very hydrophobic, meaning that they don't like to be in water, and our cells are mostly water. And so they have to figure out how to get into a membrane without having other bad things happen. And so we study the processes that are involved in that. The other thing that we do, and maybe an area that we're recently pretty excited about, is thinking about how we can design new therapeutics to target bacteria. And so here we're talking about antibiotics. And so the idea is that if we could understand how some essential components of bacterial cells work, then we can use small molecules or other tools to actually prevent those molecules from working or those proteins from working. And so by understanding the structure and sort of how things interact and then predicting function, we can then design new drugs or new other therapies. And right now we're really excited about the fact that viruses of bacteria, which are called phages, some of them actually make small peptides that really act like antibiotics. And so for the first time, I think we're really able to go through and understand how those work. And we're hoping that those will eventually develop to new kinds of therapies or new paths towards therapies.
Yeah, that's super interesting. So what were some of the biggest challenges that you faced in research and how did you overcome them?
You know, research is always challenging. So, it's hard to pick the biggest. The hard part about research is the fact that you're working on things where you you've done a lot of homework, right? You've read a lot of papers, you've educated yourself to a problem, you've been working on it for a while so that you feel that you're an expert. But the reality is you're now trying to take all of that to the next step where you don't actually know where the answers are. And so, thinking about it from that perspective, here's my hypothesis. I believe it should be true, but I've got to prove that it's true. And sometimes it's wrong. And so, when you're operating at that, sort of forefront of knowledge, then, failure is just a complete part of that. To be a good scientist and to be challenging our understanding of how things work means that there's a lot of failure involved. And so you're sort of constantly discovering that you don't know how things work and the assumptions that you made were wrong or the assumptions that were made before are wrong.
Yeah, okay. And then what do you enjoy most about your research?
As a structural biologist, I think the real joy comes from the fact that we have these moments where we get to see macromolecules engaged in a process for the first time ever. And so, you get to sort of see something and realize that, no one in history has ever seen that before. And, that's pretty exciting, right? It's pretty exciting to have those moments where, for us, we're driven by questions. And so answering those questions is a really profound and exciting moment where you can say that with confidence. But then to sort of sit there with it for a little bit and realize that, this is something that is important for humanity to understand. And you're the first person to actually, create that knowledge. I mean, there are a few things like it.
Yeah, that sounds like an awesome experience. And I know that you mentioned your PhD advisor who won like the Nobel Prize, but
Yeah.
which person or mentor in your field has made the biggest impact on you?
I think you cultivate mentors all the way along if you're doing it right. And I think, going back to elementary school, I can think of my science teachers and up through, high school and into college, people who've had impacts on me every step of my career. My family background it definitely has zero science in it. And so I had to learn a lot about what it means to be a scientist and learn about myself along the way. And I think the real benefit is that there were people who took a chance on me, but also, saw things in me I didn't see in myself. And some of that just because I didn't know to see it in myself. And so part of that is really just, finding the right people in your life to allow you to be able to do that. My PhD advisor, Venki Ramakrishnan, obviously meant a lot in my career and really helped shape me as a scientist and showed me one path to being a great scientist. During my postdoctoral work, I worked with Tom Rappaport, who was a biochemist and cell biologist, and Steve Harrison, who was a biochemist and structural biologist. And they both are pretty different kinds of scientists in many ways, but they both gave me really, I think, useful perspectives about thinking about how I navigate my career. But that said, I'm inspired by my students. I'm inspired by, we had a high school kid in my lab this last summer, and her last presentation just blew me away. And I was just thinking, wow, I'm so impressed by, what the potential is for people like that who I think are really just excited and engaged.
Yeah, okay. And then were there any pivotal moments in your research career that made you change topics or want to pursue a certain topic?
I think it's interesting. So for me, structural biology wasn't something that I knew about in college. And I didn't learn about it until my senior year when we had a course that was on, topics in biochemistry. And so structural biology at the time, it was a field. It wasn't a very common thing for universities to have. That's very different now. Now, like, structural biologists are everywhere. But 30 years ago, you were sort of in a position where I was sitting in this class and heard about structural biology for the first time. And once I heard about it, I was like, okay, I don't really understand what this entails, but this sounds like what I want to do. And so, that really kind of set me off from that moment because from that point on, I really discovered, this real joy in this field that I've never lost. And so I'd say that's, one aspect that shaped things. In my own group, we've gotten into research areas in very funny ways. And sometimes, there's a clear path to how we got there where, some person joins the group or somebody comes to us with a really interesting problem that we hadn't thought about before. But other times it was just serendipity and just sort of casual conversations and talking to peers or students or postdocs. And you suddenly say, that's a really interesting question. Do you think we could answer that question? Right. And just this casual conversations. And then that sets you on a whole nother path. So I think, following your curiosity is part of the joys of being a scientist. And I think as an academic scientist, you have the opportunity to do that, really actively. The reality is there is a work component too, because you have to, see the answers through to the end and prove them to, as well as you can. But you also, have to get things funded, you have to publish, you do all these other things that are kind of real work associated with a career.
Yeah, okay. And then where do you see your research field expanding to? So, like, what are some interesting topics that can be further explored within the next 5 to 10 years?
Yeah, so like when I was saying with our interest in sort of how these viruses kill bacteria, I think the work that we've done so far and building on others' work has really shown that there's lots of opportunities for leveraging viruses because they're essentially an infinite resource for potential peptides to target lots of different parts of the bacterial cell. And so, these viruses evolved specifically to figure out how to kill a bacteria. And in many cases, we know that they have peptides that will kill the bacteria, but we don't know how the peptides work. And so that's pretty exciting, right? Because there's mysteries there. And I think the hope is that that'll teach us new biology, but more importantly, it'll teach us new potential targets for therapies. And that's really the key right now for development of antibiotics is to identify, new options for things that you can target with drugs.
Yeah, that's really cool. So, going back to like your journey through research, where did you start? So, like, what were some basic lab positions or internships that you had?
I didn't know anything. I really come from a place of ignorance. In some ways, I went to Virginia Tech as an undergrad, largely because I was from Virginia. And it was a state school that offered me a scholarship and had, from what I could tell, was a really strong biochemistry program. I was lucky because I thought I'd like biochemistry. Turns out I fell in love with it when I started. And so I guessed right on that one. And so then I, joined a biochemistry program. But then my academic advisor at some point, asked me if I'd be interested in doing some research in his group. I thought that sounded pretty cool. And so I did start doing some research in his group, and that's when I found out that I love doing research. It was just so much fun. And so, I mean, during college, I actually had to work myself through college. I had a scholarship for most of my tuition, but I had to pay rent and all the other things. And so balancing volunteer research with a job and everything else that I was doing. But in the end, that really, positioned me quite well, I think, for understanding, what it was going to take to be good at the graduate level. And so when I got to grad school, I think I was in pretty good shape. But from there on, I don't know if it's a traditional path, but I certainly followed a path of sort of, PhD to postdoc to faculty position.
Yeah, your whole journey is like really inspiring. It's cool how like you put yourself through college, just like using scholarship money and like your own job. I think that's really amazing.
Yeah.
And then these questions are more towards like high schoolers. So,
Mhm.
how did you find your passion, and how would you suggest a high schooler try to find their own passion for research?
I think in high school that one of the keys, is just to be curious, right? And you're not going to figure it all out in high school. And you don't have to. There's a lot of life ahead of you. And, by the time I got to college, I was clueless, and I didn't know anything about college. I didn't know anything about being a good academic. And I had to learn a lot of things because, through my education, I guess I was fortunate enough that I was able to kind of skate through most classes through high school because, I was in environments that were challenging. But I just felt like I didn't have to work super hard, and I certainly didn't have, a support network where my parents were pushing me. And so that meant by the time I got to college, I was just completely unprepared for college. And so college hit me like a brick. And so it took me a little while to figure that out. Not that I ever completely did, to be honest with you. But the point of all of that is to say, it's a different world now, right? Getting into college is much more competitive. Here at Caltech, the average undergrad who gets the Caltech has already done a research project. It's a little crazy. And so, I don't know that my experiences, sort of naturally, dovetail into what others' experiences might be. But I do think, you're only young once, live your life. If this is the right place for you, you'll find it eventually. Navigating, the things that you're passionate about, finding out that you've got a love for literature and a love for chemistry. I mean, those are okay things to have a love for both of those things. And, at some point, you have to make a decision which path you're going to go on. But that's not a choice you have to make tomorrow. And then certainly, most of the time when you pick universities for a variety of different reasons, but when you pick the kinds of schools that you want to go to, as long as they have the opportunities for you to sort of, have options for the path that you want to be on, then you'll be fine.
Yeah, okay. And then what kind of skills do you think a high schooler could develop, even if they don't have access to a wet lab?
There's lots of opportunity to sort of dig into the science, now just through virtual, and, for virtually educating yourself. And so I don't think you have to feel completely disconnected from science. I think there's lots of opportunities actually, with this sort of era of big data and AI where there's all kinds of tools that are just freely available for you to have access to. And so learning about how to interact with things and engage in things, I think is pretty amazing. One of the things that we play around with every day, but there's no reason why high school students can't play around with it, is AI tools for protein structure prediction. They're completely easily accessible now. Things like AlphaFold 3 and Chai Discovery, CHAI Discovery. These are actually sort of really interesting tools that have sort of opened up the world because they actually do a pretty good job of predicting a lot of different kinds of things. Things that it used to be my job as a biologist to discover. Fortunately, the things that I care about are sort of the next generation problems that, it'll be a while before AI can solve those problems. I hope it does eventually. But what it means is you can start playing around with genomes and start asking these kinds of questions. Thinking about things like understanding what a complex protein that you're interested in for whatever reason looks like, and then maybe trying to understand what it interacts with and things like that. I mean, there's lots of sort of opportunities at that level. And those don't require wet lab at all. So, I think from the perspective of what can you do from a wet lab, that's a little hard to think about sort of how you can do that, but there's no reason why you can't do experiments on your own. And so I think just leveraging the tools that we have now, I mean, your generation's going to have a wild ride because, you're really getting educated in this AI world, which, my generation has to figure out too, but your generation, this is the only thing you're going to know. And so, what does that mean for sort of how we do things? And I think, AI is a great teacher. It's not absolute teacher, right, in terms of getting everything right. But I think you can you can even ask questions, if I wanted to do my own, microbiology lab at home, I'd be curious to know what AI might come up with as a way to sort of help you figure that out. I think there you could do some really interesting stuff.
Yeah, okay. I'm sure that you probably get a bunch of like emails from high schoolers or undergrads asking to join your lab, but like,
Yeah.
how do you select someone out of that? So like, what makes someone stand out?
I think a thoughtful email helps. I think a realistic email helps, So I'm not super keen on free labor. And so, and mostly just because I don't think it's fair to anyone to do free labor, right? But I understand, I'm not inclined to pay high school students to do work. And conversely, I think you don't necessarily care whether you get, compensated. So, I understand that. But so because of that, then I think, a student trying to be in my lab is, emailing me from San Francisco or something, we're in Los Angeles, right? It's a little wild, right? So to imagine they're going to put themselves out as a family to try and figure out how to work in my lab seems a little over the top. So, I typically don't get super excited about that. Most labs don't take that many high school students, so you kind of have to just be the first one to get noticed because once I've accepted somebody, if I do that year, there's not really room for a second student, sort of weird aspect. So I don't know that there's anything that particularly stands out except for the fact that I think a student who has really had to take the time to sort of think about what we're doing and reaches out in sort of a way that seems feasible for them to work in my lab without a heavy burden, that seems like someone not worth having a conversation with. Somebody who's got to, commute 30 miles to get to my lab. It's just, man, I don't want to disappoint you. I get it. I mean, there's the most important thing I think, 'cause it's hard work, identifying labs that you're excited about. But I think if as you do that, and then on top of that, I think, being open, if sort of, at the end of your sophomore year and you're reaching out and saying, hey, do you have any opportunities? And if someone says, no, we don't have anything this summer, then, well, what about next summer? Right. And, when's a good time to reach out and talk to you about possibly doing something next summer? Because, then maybe you're ahead of the game and the timing's probably a little better in terms of your own experience.
Yeah, okay. So it's really just being like realistic, but still like dedicated and like interested in it.
Yeah, most of the time the answer is going to be no. You're not going to be surprised by that. And the reason is multi-level, but the biggest thing is, as a high school student, what do you bring to the table for us? Right. And we don't expect actually our high school students to bring anything to the table necessarily. And so then I guess the question is if I'm going to be investing, our resources and our time as a resource, is that going to be well invested in you? Right. And so if that translates into you sort of figuring out who you want to be for the rest of your life and maybe getting a science degree and sort of pursuing something in that direction and sort of setting you on a path, I think that's a good investment, right? So it can just be that, but it has to be something where you have to really, show that there's a commitment and a dedication to you valuing our time. That's everywhere. I think that's a big part of it. The high school students who are, I think, you get disappointed, right? Who clearly just, are worried about their college application. They don't want to be in your lab because, I mean, they're happy to be in your lab, but they didn't specifically choose your lab because they're excited about the science or the things that you're doing. They're just trying to find a lab so that they can, get that additional part of their college application filled out. That's not super exciting.
Yeah. And then what type of traits do you think define a great researcher? And like what type of traits would you look for in a potential lab member?
Curiosity is the first and most important thing, right? I mean, you really have to be driven by a curiosity because, curious people aren't satisfied with not finding the answer and push hard to try and discover things. I'd say that's a big part of it. And curiosity means that, you dig into questions, right? And so you've thought deeply about sort of why we're having a conversation and what you're excited about and what you can learn more about. So I think there's that aspect of it that you're sort of driven by that curiosity that make you push and work hard in the space. The other thing is just a real commitment to the time that you're going to invest, right? Because your time should be precious to you too. And I think, choosing to, come and work in my group, there has to be, drive on your part, right? Because I can invest my time in a lot of people, and so if I'm investing my time in you, I want to know that you're really valuing yourself and your time in a way that's going to help you achieve with this. Because if not, then there's other people in line who can benefit probably more from that experience.
Yeah, okay. And then just for our final question, what kind of tips would you give to a high schooler who's just looking to go into research?
I think you can overthink these things now. I think the thing is, really engage in your curiosity to start thinking about what kind of science excites you the most. I think people have a tendency to get a little caught up in sort of the experiences that they've had as a way to sort of shape where they think they want to be. And by the time you're in high school, your idea of a scientist is not well formed. And moreover, I think you just you haven't seen enough things to know what you're really passionate about. You have so many opportunities through the web and podcasts and other things to really start exploring different kinds of science to sort of see, this is an area that really sounds exciting to me. I think you should be open-minded, for the rest of your life if you're a scientist. So I don't think there's a point where you have to say, okay, now I figured it out, because you may discover at any point in time that there's something else that you want to be invested in. But as you're navigating it, finding the thing that really excites you as a problem to solve, that's really critical to being successful. You know it when you find it, and that's, I think, a sort of a weird way to frame it, because how do you know you're ever going to find it? And I don't know, right? But I think the chances of finding it by exposing yourself to more stuff is really important. And so I think that's the thing that you can kind of keep in mind, is just try to expose yourself to as much stuff as possible. And I think, as you do that, that will help you figure out what path you want to be on.